Welcome back to the Deep Dive. Today we are taking a massive stack of research papers on microbiology, neuroscience, and even oncology. And we're tracing the story of one single tiny molecule that connects them all. If you've ever seen the movie Lucy, you might remember the fictional drug CPH4.
Oh, right.
The one that's supposedly unlocked, 99% of the human brain's potential, granting superpowers, omniscience.
The ultimate brain hack. And this is where reality gets, just as strange as science fiction.
Go on.
CPH4, the pathway around it, 6-carboxy 5,006,7,8 tetrahydropirin, is absolutely real.
Way.
It's not a miracle drug, but it is a crucial precursor molecule made by certain bacteria, like the ones living in your gut right now.
So our mission today is to follow that real bacterial molecule, CPH4, all the way to its final form, a hypermodified nucleoside called Q-azine, or just Q.
Right.
We are going to explore how this single modification, which we can't even make ourselves, is salvaged from our microbiome.
Yeah.
And how it literally controls the efficiency and the accuracy of our most foundational biological process-making proteins.
Exactly. We're diving deep into the microscopic editor that dictates everything from a bacteria's ability to shrug off antibiotics to how our neurons fire and how our memories are laid down.
It's an incredible link, then, between our microbial neighbors and our own neurological health.
A completely systemic link.
Okay, so let's unpack this. We've said Q-azine, or Q, is a hypermodified nucleoside, but where does it actually live?
It lives in transfer RNA, or tRNA, specifically at position 34, which scientists call the wobble position.
For listeners who maybe don't think about RNA on the weekend, what is tRNA's job, and why is that wobble position so critical?
Okay, so think of the ribosome as the protein factory. The mRNA is the blueprint.
And the tRNA is the translator, the delivery truck, maybe?
Exactly, it's the delivery truck. It matches the code on the blueprint, the codons, to the right building blocks, the amino acids.
Okay, so where does the wobble position fit in?
The wobble position is like the flexible hitch on the back of that delivery truck.
Yeah.
It allows that tRNA to recognize and latch onto several slightly different versions of the code.
So it gives the system some efficiency, a little bit of flexibility.
Little margin for error, yeah. Now, Q comes in and modifies that flexible hitch. It fundamentally regulates the efficiency and the fidelity, the accuracy of the whole process.
So Q is like the quality control officer.
A quality control officer making sure key amino acids like tyrosine, histidine, and aspartic acid are slotted in correctly and at just the right time.
So it's not just about accuracy, it's also about the pacing of the whole assembly line.
Precisely. And here is the profound twist, the part that forces us to look at the microbiome.
Right.
Eukaryotes, so every cell in your body. We can't synthesize Q from scratch.